Okay, thank you very much for the invitation, first of all, and thank you very much for the kind introduction. And you will hear in a minute why I'm a little bit afraid of being in that audience. You will encounter the far other end of what we have heard before. I'm a microbial physiologist, and we are interested in lifestyles of bacteria and archaea. Organisms you have just heard, organisms like Desulfovibrio, like Pyrococcus, like Methanococcus, and I will add two more to that list of exotic bacteria today. So we are interested in lifestyles and energy metabolism of these unusual bacteria, and today I'm going to talk about a group of bacteria which is called the acetogenic bacteria, so anaerobic acetate-forming organisms. And they all grow by conversion of CO2 to the end product, which in that case is acetyl-CoA. And that slide compiles all the 7 known routes of CO2 fixation in nature. And if you look at the energetics, you see, for example, Kelvin cycle requires 3 ATP per CO2. And so on. And if you go here to the Wood-Ljungdahl pathway, that does not require ATP for CO2 fixation if coupled to membrane-bound reaction that I'm going to talk to you in a minute. So that is the least energy costly way of CO2 fixation in nature. And that's the reason why there are 3 major groups of bacteria: methanogenic archaea, sulfate reducer that we already heard about, and acetogenic bacteria use that pathway to reduce CO2 to the end product that can be methane or that can be acetate. So I'm going to discuss with you today the pathway of CO2 reduction and its coupling to energy conservation. And we've learned about high resolution. I have learned, you all know that. I have learned about high resolution structures of metal centers in hydrogenases. So this is the high resolution structure of a pond. The pond has a duck here, and it has a fish here, and it has a sediment. And the sediment is strictly anaerobic. And this is a place of action where our bacteria live. And most of the bacteria that were named today are actually anaerobes. And the anaerobes live here in the sediment of, for example, that pond. And in that pond, we have an anaerobic food chain in which polymers are degraded via monomers to acetic acid and further on to methane and CO2. And this can easily be visualized by using a vent here and collecting the gas and then taking advantage of the fact that methane burns. You can easily demonstrate that methane evaporates from the sediment of the pond. So the pathway that these bacteria use is the so-called Wood-Ljungdahl pathway in honor of the 2 people that actually established, identified, and characterized that pathway, which is a linear pathway of CO2 reduction that has 2 branches. In one branch, CO2 is first reduced to formic acid by a formate dehydrogenase. Then the formate is bound to a cofactor, tetrahydrofolic acid, subsequently reduced to a methyl group. And then in the second branch, the carbonyl branch, the second molecule of CO2 is reduced to carbon monoxide, enzyme-bound carbon monoxide. And the carbon monoxide, together with the methyl group and CoA, is condensed on the key enzyme of that pathway, which is the CO dehydrogenase acetyl-CoA synthase to acetyl-CoA. And since all of you in the audience are interested in metalloenzymes, that is also a nickel-containing enzyme, the H2ase, and there has been much work on how that enzyme works on a molecular level. So if you look at the pathway and look for the energetics, then you see that 1 ATP is investigated over here to activate formate, and 1 ATP is gained here in the acetate kinase reaction. So that makes overall 0 ATP that is produced, but there's also 0 ATP that has to be invested. However, in order for these bacteria to grow by the conversion of CO2 to acetate, there has to be additional energy conservation. And how that is brought about, the mechanistics of energy conservation, this is what I do actually since my diploma work in the mid-80s. And the energetics of the overall reaction is shown here. The production of acetate from hydrogen CO2 goes along with a free energy change of only -95 kilojoule per mole of an acetate. if you take molar concentrations of hydrogen that you never encounter in nature. So if you take hydrogen concentrations that people actually measured in anaerobic ecosystems, which are well below 1 micromolar, you end up with delta G values of around 20 kilojoule per mole of an acetate, which would give you less than 0.3 mole of ATP per mole of an acetate. This is what we call microbial life at the thermodynamic edge or at the thermodynamic equilibrium. And our group is interested in the question, how do bugs survive with only that little increment in free energy change? And that can be even worse. So microbial ecologists tell us that the biomass, for example, in the Bay of Aarhus in Denmark, doubles every couple of 100 years. So the delta G values are even much, much, much smaller. Nevertheless, life is sustained. And our group, as I said, is interested in figuring out how these life-sustaining processes work. And I give you two examples today, two of our main organisms that we work on. One is called Acetobacterium woodii, that's a mesophile, and the other one is called Thermoanobacter kiwi, which is a thermophilic organism, grows optimal at 66 degrees. What you see here is the pathway of CO2 reduction to acetate, but now we have a surrounding here, we have a frame, that frame is a cytoplasmic membrane. And now I'm going to demonstrate to you how additional energy is conserved. First of all, we need a hydrogenase to activate the hydrogen. And we all know about the function of hydrogenases. And in both organisms, we have electron bifurcating iron-iron hydrogenases. And it's a pity that I just learned that Jan Schuller, my collaborator from the University of Marburg, who was supposed to give a talk later this afternoon, on the structure of electron bifurcating hydrogenases from woodii and kiwi that we actually solved recently. And we did quite a bit of site-directed mutagenesis to figure out the mechanism of electron bifurcation. But as I said, I just learned that 10 minutes ago that he will not be able to talk about that. So if I would have known, I would have been able to show you a couple of slides on the structure of the electron bifurcating hydrogenase, because that was mentioned already in the talks before. So we need an electron bifurcating hydrogenase, and then in the second step, in the second step, we need energy conservation. And we have two respiratory enzymes so far in the acetogens. These are the only respiratory enzymes found. Some acetogens like woodii have the so-called RNF complex that oxidizes reduced ferredoxin and reduces NAD, and that is coupled to ion export out of the cell. In Acetobacterium woodii, that's a sodium gradient that is established. Other acetogens that have RNF complexes do produce proton gradients. So there is a diversity, whether there's a sodium or proton gradient established. Thermoanobacter kiwi now has the second hydrogenase in its metabolism that is important, is the energy converting hydrogenase, the ECH complex. That enzyme also uses reduced ferredoxin, but it reduces protons to hydrogen gas and it extrudes protons. For physiological reasons that I'm not going to discuss here in detail today, in the cell, the enzyme works in the reverse direction. Therefore, it's written in the reverse direction here to reduce ferredoxin. So the potential established by these enzymes is then used by an ATP synthase to generate ATP. And there's also a big diversity in the enzymes involved. That is what we have done the last 20 years. The enzyme in Acetobacterium woodii uses a sodium potential. It has a very unusual C subunit, the rotating C ring in the membrane that transmits energy, electrochemical energy into rotation of protein subunits. And in Thermoanobacter kiwi, the driving force is not a sodium, but it's a proton gradient. So that is, in very short, the summary of 20 years of work on how energy is conserved by respiratory enzymes that generate the potential that drives the synthesis of ATP. And ferredoxin is generated from hydrogen by an electron bifurcating hydrogenase. The reduction of ferredoxin with hydrogen is highly endergonic and only made possible by the simultaneous reduction of NAD, and that energy is transferred in the hydrogenase to reduce ferredoxin at the expense of hydrogen. So I'm going to concentrate today on the first reaction in CO2 reduction, which is the reduction of CO2 to formate. If you reverse the system, then you have a formate dehydrogenase, and this conference is called metalloenzymes. All of you are aware of formate dehydrogenase that are all metalloenzymes. So we looked for these formate dehydrogenases in these two organisms for physiological reasons that I'm not going to be able to discuss in detail, but in very short, if that reaction here would be driven by ferredoxin, there would be no ferredoxin left for the pump, and there would be no energy conservation. So we had to look for the physiological electron donor, which cannot be, for physiological reasons, ferredoxin. So we looked for that, and here you see the energetic potentials. The electron donor for CO2 reduction cannot be NADH. So you all know that formate dehydrogenase is coupled to NAD, but these are all formate dehydrogenases from aerobic organisms that are in the cell employed in formate oxidation to CO2, and then NAD is reduced. But if you go into the direction of CO2 reduction, NADH is, for thermodynamic reasons, cannot be the physiological electron donor. Physiological electron donors can be NADPH, ferredoxin, but ferredoxin should not be possible for physiological reasons, and it could be hydrogen. And indeed, there is a zoo of different formate dehydrogenases in acetogens. We have formate dehydrogenases in other acetogens that are coupled to ferredoxin. We have electron bifurcating formate dehydrogenases. Our group itself has studied one of these enzymes. And we have electron bifurcating formate dehydrogenases that are coupled to another electron bifurcating enzyme, an electron bifurcating hydrogenase. And the group of Tower and also our group has studied these unusual formate dehydrogenase-hydrogenase complexes in some detail. However, the direct involvement of hydrogen in reduction of CO2 had never been described. And we had a first look at the genome sequence of our major pet, the Cetobacterium woodii, and it turned out that we did find a formate dehydrogenase gene, but this formate dehydrogenase gene was always hooked up to a hydrogenase gene. And in between, there were 3 genes that were encoding small electron transfer subunits, and that brought us to the idea that maybe the formate dehydrogenase is directly coupled to a hydrogenase via 2 small iron sulfur proteins. And Kai Schuchmann in my group, he started to purify the enzyme, and that turned out to be quite successful. He purified the formate dehydrogenase activity, and that formate dehydrogenase catalyzing activity had the expected subunits FDH, F1, 2. So there's a second formate dehydrogenase. One encodes selenocysteine, the other one does not. So we do find both isoenzymes in our preparation. We do find the hydrogenase, and we do find the small iron sulfur containing subunits. Of course, we then looked for activities, and the first thing was obviously to see whether the CO2 reduction can be driven by hydrogen oxidation, and indeed it does. You see here the activity coupled to a commercial formate dehydrogenase that then reduces NAD. You see reduction of NAD upon addition of hydrogen. So the system also works in reverse. It oxidizes formate to CO2, and protons are reduced to molecular hydrogen, and molecular hydrogen is evolved. That's a very important aspect, because if you take other formate dehydrogenases, they're all biased into the direction of hydrogen production. For our bugs, they do grow on hydrogen and CO2, but they also grow on methanol or methylamines or on formaldehyde. And under these conditions, physiologically, the formate dehydrogenase has to go the other way. It has to produce hydrogen. So the enzyme present in these bugs should be good in hydrogen oxidation, but good as well in production of hydrogen. So this is what the enzyme is. So the catalytic activities of the purified enzyme, which we call the hydrogen-dependent CO2 reductase, or in short H2ase, are compared here. Here's the enzyme from Thermoanobacter kiwi at 60 degrees. You see the turnover number here, 9 million. The one of Acetobacterium woodii, the bug that grows at 30 degrees, is 140,000, and you see the formate dehydrogenase containing the formate hydrogen lyase containing E. coli system. Also can do that, but quite lousy with an activity of only 1,800. So these enzymes, the HDCR enzymes, are orders of magnitude better than any biological catalyst, and they are also orders of magnitude better than any chemical catalyst for reduction of CO2 with hydrogen 2-formic acid. We then did quite a bit of work on subunit interactions, on cofactor content, and this is what I call a molecular hallucination. You have a gene sequence, you do some biochemistry, and then you can come up with a cartoon. The cartoon shows you here on that side the formate dehydrogenase, here on that side the hydrogenase, which is an iron-iron hydrogenase. Here's the H cluster. And then we have strings that we postulated to be strings of iron sulfur centers that connect both catalytic subunits. And that enzyme has now been purified from Acetobacterium woodii, but also from the thermophile Thermoanurobacter kiwi by Kai Schuchmann here on the left and Fabian Schwarz here on the right. First, I'm going to discuss with you the application. We were talking about in the break what is the future of hydrogenases, and for me as a microbial physiologist and biotechnologist, the future of hydrogenases are clearly in the production and consumption storage of hydrogen. You all know that we have a poly crisis at the moment, a poly energy crisis. And we have to find ways, for example, how to produce, on the one side, hydrogen, but the technology and politicians go to green hydrogen that are not produced by microbes. But we also have to solve the problems in hydrogen storage. And many chemical engineers go for reduction of CO2 with hydrogen under production of formic acid. And formic acid is a so-called liquid organic hydrogen carrier, which is liquid, non-toxic, and can be transported over long distances. That has tremendous advantages compared to hydrogen gas itself. So therefore, Fabian Schwarz developed a whole cell system in which we just have the HDCR enzyme, and it is decoupled from the rest of acetate formation, meaning here there's no acetate formation anymore. And if we decouple that, the enzymes work as ideal catalysts for the whole cells, work as ideal catalysts for reduction of CO2 to formic acid, and it's quantitatively converted to formic acid, and formic acid is excreted, as you can see here. Over time, the whole cell system produces formic acid, and it does it the other way around. It oxidizes formic acid back to hydrogen. The equilibrium constant for that reaction is 0.98, so it's well in equilibrium, and the enzyme and the cell system can be used to make formic acid from H2CO2, but also to oxidize formic acid back to hydrogen and CO2. So that was developed, and if you compare the activities, the specific formate production rates from hydrogen CO2, here in the bottom of that legend table, you see the thermophile with 152 millimole per gram cell dry weight in hours. And if you compare that to our mesophile with 22, that is much better. And if you go to E. coli under normal conditions, that is lousy with only 0.1. But there is a work presenting data with E. coli under high pressure, very high pressure. Under these conditions, E. coli also produces formic acid with a rate that is comparable to the one in Acetobacterium woodii, but still much lower than the one in the thermophilic organism. So that system can be used to store hydrogen in the form of formic acid at the place where it is produced, in areas where there is lots of sunshine, and then it can be shipped to the place of action, maybe Northern Europe, and there the liquid formic acid can be oxidized to hydrogen and CO2 again. And if you follow the news, for example, here in Germany, we have now finished the first station where we can actually handle LNG gas, and the same station can also handle liquid hydrogen. So the other dream that we had was to have a hydrogen storage device that can be used individually in houses, in households, where we have a day period, and during the day period we have solar energy driven here by light, and that solar energy drives electrolysis of water, and thereby hydrogen is produced. And then we take our bacteria, feed them CO2, and then they produce formic acid. In the night period, 16 hours, then the formic acid is automatically oxidized to hydrogen back and CO2 recycled, and the hydrogen is then used as energy source for various devices. So that was the idea, and we tried that very hard. We set up a fermenter to do these kind of experiments, and the other thing was strain design, which was very important. I told you that formic acid is the intermediate of that pathway, so we had to generate a knockout mutant, and that knockout mutant was done by Yim-Young Moon in my group, and he knocked out that key enzyme here, the methylene tetrahydrofolate reductase. And under these conditions, CO2 is quantitatively converted to formic acid, and formic acid is extruded. So having done that, we were now able to use our fermenter to feed it with hydrogen during a simulated day period of 8 hours. And you see during that day period of 8 hours, formic acid increases. And this here is a blow up of that period. Here you see during the day period, formic acid is produced. And then at night, when we turn off hydrogen supply and supply the system with nitrogen, formic acid is then reoxidized, it goes down, and hydrogen is actually produced. And you see, we can do this in one fermenter for a reasonable time of over 360 hours. And then with time, it is degrading, which is due to the instability of these cells. These cells are non-growing cells. These cells are so-called resting cells that only have energy metabolism, but no anabolic reactions anymore. So with that, we were able to actually generate a biobattery. And this biobattery that made big waves in newspapers. It was published actually this year, and we are still working on it to scale it up to a commercial scale. So now I would like to switch gears and come to the structure of the enzyme. And for determining the structure of the enzyme, we needed a mutant. And I told you about the unusual bugs, and you heard about Desulfovibrio and Pyrococcus and Methanococcus. And for most of these systems, there is no genetic system. You heard the talk by Mike Adams at the beginning, and Pyrococcus, that is one of the few organisms for which Mike Adams and others developed a genetic system. And we did the same for Acetobacterium woodii and for Thermoanurobacter kiwii. So we established genetic systems, and these systems helped us to do structure function analysis of proteins. So if somebody in the audience is interested in structure function analysis, for example, by site-directed mutagenesis in any of your favorite metalloenzymes that can be produced in a thermophile or in a mesophile, we can offer help in actually producing proteins under strictly anaerobic conditions in these bugs due to a genetic system. And here you see Zobi Yain. She did the PhD with me, and she's now a postdoc in Chris Greening's lab. So she left damn cold Germany to go to warm Australia, and she generated a mutant of Thermoanurobacter kiwii, which is devoid of, here you see the genes encoding the H2ase enzyme. And that strain does no longer grow as expected on hydrogen and CO2, as you see here. And that strain was then used by another enzyme, Helge Dietrich, to produce formic dehydrogenase expressed from a plasmid that replicates in this thermophilic anaerobic organism. And by using that plasmid-based system of protein production, Helge was able to actually do site-directed mutagenesis on the enzyme. And that helped a lot after we developed the structure to figure out function of the enzyme. So that is a cartoon that we had already from structure function analysis that we did in our laboratory. As I said before, format dehydrogenase, hydrogenase, and both are connected by these two small iron cipher proteins. And what we also saw from the first preparation that we had already in 2016, electron micrographs taken by Janet Funk here at the Max Planck Institute for Biophysics in Frankfurt, we did see that the enzyme formed huge filaments. But at that time, we were not sure whether this is of physiological relevance or whether this may be an artifact. But what we saw is that when we disrupted the filaments and shortened the filaments, the activity that we measured went actually down. And then we were very lucky to meet Jan Schuller, which is shown here on the right-hand side. I will do this again. On the right-hand side, that is a structural biologist from the University of Marburg, as I said, intended to give a talk later on, but obviously got sick and can't do that. And this is our student Anush Kumar. And they were brave enough to study the structure of that filamentous protein by cryo-electron microscopy. And what you see here is that structure. You see a wire in the middle, and you see in blue the hydrogenases, and you see in green the formate dehydrogenases. This is a short filament. And the filaments are made by repeating units that are shown here in the middle. That's the formate dehydrogenase. These are the small iron cipher proteins, and then it has two hydrogenase subunits. And these repeating units actually make a filament. And what we saw was that there were in these small proteins, these small electron-carrying proteins P3 and P4, a C-terminal domain that is important for making actually that wire system. And then we took advantage, as I said, of our mutagenesis system, and Helge Dietrich knocked out that C-termini. And what you see here, when he did that, knocking out the C-termini, that gave mutants that were no longer able to efficiently produce hydrogen from formic acid. So this is around 10%, 20%, this is around 6% of activity. And what you also saw was that in these mutants, the length of the filaments was drastically reduced. So length of the filament goes along with increase in activity. And we actually also wanted to see whether there is electric connectivity in that filament. And what you see here is the nanowire, and here you see the iron cipher centers. This is actually already, for me as a microbial physiologist, a very high resolution structure, because I do see the differences in distances in different iron cipher centers here. And we knocked out always these iron cipher centers, which are here in brackets. And when we do that, the individual activity of the unit measured with formate production from CO2 or with methyl viologen reduction with hydrogen or formic acid is constant, but the overall activity decreased, the size decreased dramatically, and the overall activity decreased dramatically. Here's a wild type, and here you see when we knock out that iron cipher center that bridges the repeating units in the nanowire, there is very little activity. And here you see the advantage that we have in our system, because we can do these genetics in the host. You see that the mutant cell does not longer grow, but the where the HDCR is completely deleted does no longer grow. But in this mutant version, growth rate is slower, but cells actually grow.
Professor Mueller?
Yeah.
We're running a bit over time already. So
okay,
if you can finish in about 5 minutes, that would be perfect.
Yes, that's doable.
Great.
So this is a picture of the entire long filament. You see the nanowire in the middle. And again, you see the hydrogenase that twists around the wire, and on the other side, so you see the formate dehydrogenase. And here you see the connecting iron cipher centers. So we then submitted that for publication, and the reviewers were arguing that this may be an artifact and we should be trying to demonstrate that the enzyme forms long filaments in vivo inside the cell. So we teamed up with Ben Engel and his team, formerly in Munich, now in Basel, and they did cryo-electron tomography on the system, which I'm showing you now. Now you see a view inside the cells. This is the Thermoanobacter kiwi cell. We see invaginations that we had not seen before. And now you see in blue the ribosomes, and in yellow you see the H2ase enzyme. The H2ase enzyme seems to originate from the cytoplasmic membrane that you can see here. And what the H2ase enzyme does is, what you see here is, this is one filament. These filaments are turned around each other to make supramolecular structures, which we call bundles. So what you see here is, this is a bundle of filaments of the H2ase. And now we move into the cytoplasmic membrane. And now for comparison, in blue, the ribosomes again. So why that enzyme is such a fascinating filamentous structure is at the moment completely unknown, but of course opens new areas of research that we are at the moment exploring. So with that, I'm already at the end. I hope that I've convinced you that the H2ase enzyme is the most efficient catalyst for binding hydrogen and CO2, and this is where I see a future of hydrogenases. In biological hydrogen storage and release, the H2ase forms filaments with heterohexamers as the minimal functional unit. Protein connectivity and electric coupling is observed in that filament, and it's important to maximize enzymatic activities. And in vivo, the H2ase filaments are bundled together to form large ring-shaped superstructures. And these superstructures are membrane associated and contain thousands of iron-sulfur clusters. I've mentioned names of people involved along the talk, and I'm happy to announce the funding, which is from the DAAD that funded Sobi, Deutsche Bundesstiftung Umwelt, and of course from the ERC, and I thank you for your attention. Thank you.